Yeah, I welcome you all for this talk. Today we have Professor Balaji Jayaprakash. He's going to give a talk on mapping the memories in brain, and he works extensively in microscopy imaging, and that you've already seen here, right? I'll give a little brief of Professor Balaji. He did his postdoc, PhD at TIFR, TIFR probably, followed by a visiting fellow. He was visiting fellow also there. Then postdoc at Well Medical College, Cornell University. Cornell is known as one of the epicenter of microscopy imaging. It's one of the big centers for that. Followed by another postdoc. He was a researcher at UCLA, University of California, Los Angeles, from 2007 to 2011. And he, as I know him, he has a rich experience and he goes deep into the matter, which means, he's quite thorough with things as much as I've seen. And he leads a neurodynamics lab where he studies neuronal correlations in the context of learning and memory. And this is something that we're going to witness today. Real-time neuronal activity is something that he likes a lot. And the techniques that he uses are basically multi-photon microscopy, spectroscopy, virtual reality, and optogenetics. Okay. So, I guess we'll have a very good talk, and we're looking forward to it. So, Professor Balaji, if you can take it.
Thank you, Partha. And just thank you, Partha, for giving us such a wonderful to that, right? So there's no. Well, as Partha said, our lab, neurodynamics lab, we are interested in understanding how our brain forms memory, stores memory, and uses these memories later in time when you're faced in a situation that you have not faced with. One of the very, very stark features of this memory use or use of this memory is that whether we want to believe it or not, the memories that we store, right? We all would like to say that we know it for very certain, right? With this strong certainty associated with it. I know for sure this is right? There is no uncertainty associated with it. But the reality is very far from what we like to believe. Memories are just the uncertainty is very much tied to them. You cannot just say that, this is for sure. Okay? I'll do this with a small example, then come back to what we do in the lab. The uncertainty comes when you ask, hey, while you are actually taking the picture, do you remember there was a board right next to it? Yes, I know there was a beautiful houseboat that was right next to it. Now, can you tell me, was it like this or was it like that? Okay, if you ask that question right next day, probably you would be able to tell that. But a month later or a year later, if you ask that question, you have that board in this board, right? You can differentiate them. This orange and that's apple, or that's apple and this orange, right? Whichever way you like to follow. Okay? So then it's not that you're unable to say what these two boards are different or not, but your memory, you can't rely on your memory to be able to say whether it is whether it is this board or that. Okay? Now, we are deeply interested in this. Why? What we believe, and we have shown in the lab experiments too, that these kind of uncertainties shape our behavior. In fact, it shapes our behavior so much that many times when we actually make an estimate of what I should be doing, you rely on how uncertain the memory is. Okay? If you're interested in what the state means or how does it work, that's a completely different direction that we have to take it to. I'm more than happy to talk to you after that talk, but that's the general theme of the lab. Okay? So where is light coming in? Where is microscopy coming in? Where is imaging coming in? In order to understand this, one of the key factors that we need to be able to do is to be able to look at the representation of information inside the brain as it happens, while it happens. Now, how do you do this? The way you do this is first by understanding what are the different forms the memory can take. What are the different forms the information can be stored inside the brain. Those different forms ranges from the ability of the neurons, right? The neurons are the fundamental units of the brain, right? You can actually make the neuron conduct an electrical impulse. Depending on that, you say that whether the neuron is active or inactive when it conducts, and that action potential. So the firing of the neuron or the action potential reaches its action or terminals. It crosses over to think in terms of an amplifier. This is about talking about modifying the gain, right? Modifying the threshold of fire, right, of your detection. If you change your threshold, you can actually, by changing the threshold, you can make the neuron fire earlier or later. For a lesser signal or a higher signal. So that's one way of storing information. But this action potential needs to cross over to the next neuron. That happens through neuronal connections. We call it synapses. You can actually make these connections to becoming more probable or less probable. Modulate the conduction. Right? That's another way of storing memory. Apart from this, there are also molecular players that you can actually encode memory. Okay? So you can look at all of this. One of them, or maybe there is something more, where you are actually looking at the network connectivity itself. This is not something new here. This has been postulated and been kind of studied from 1800s, right? Ramanika Hall. He looked at the brain sections, and what you are looking at is the picture or depiction of these brain sections that he was looking at of a neuron, how it looks like, and through the microscope, where you are looking at that region. You see this small pockets here, right? The places where you can actually form these synapses. Okay, this end of it. Okay? Remember this. This end of it is what this is. So when the brain stores information, so that's very intriguing hypothesis. But how do you go ahead and test it? You can go train the mice, train the humans. After sacrificing the mice, postmortem of the human brain, you can actually take a section, start to count these pantas, and see has it increased. It's a hard job because you don't. There is so many different variables out there, right? And on top of it, you don't have a real control over what information has it been stored, what is it in response to. And that both of them are completely black box. In the mice, people have done this. where they're trying this. A hell of a lot of mice, right? A group of mice, big group of mice, trying them post-training and before training, same group. You sacrifice them and start reaching. You can actually zero in based on the XYZ coordinates with respect to its body. So once you do that, then you take a more or less a similar. It's hard for me even to describe, let alone doing this, right? And still there are a lot of problems in actually relating this memory in the brain. What stops us from actually looking at the brain while this process is happening? Number one, if you have to look at it, you need to be able to see at this scale, right? Here, scale, when I'm talking about, we're talking about spatial scales of few hundreds of nanometers. The biggest of them about a micron or micron that. Not only that, you have to be able to do that in a live mice or at the best, right? Live mice inside the brain that is deep inside. I mean, these structures are deep inside, but the actual memory is not. So how do you go about doing this? Some of this high spatial resolution requirements and the depth resolve imaging can be achieved with confocal microscopy, right? Because might have heard, many of you might have heard or learned about this confocal microscopy, wherein you know that you have an excitation light, you focus it through an objective lens onto a small sample here. The idea here is that once you excite the fluorescence gets collected back, passing through the dichroic. Passing through a dichroic mirror, and then what you are actually doing is you're collecting it through another lens before putting through a pinhole. The idea here is that when you have a pinhole, the fluorescence that is originating from after the focal plane, as well as that of the light coming before the focal plane, both of them can be nicely discriminated because the other guy is going to expand out because it's going to focus before and expand out and letting only a small fraction of the light going through, while the other, right? And your ability to be able to say, okay, it is coming from here, not from there, or not from here. But the problem is, the whole discrimination comes from, our whole depth information comes from your ability to discriminate in Z, right? You're throwing away light and saying, I am only looking at the light that is coming from focus, not so that is beautiful. When you are looking at the sample that is kind of semi, look at the brain. Have you, any of you looked at the brain? How does it look? What color does it look? Pale pink, correct? And if you take a section and you drain it well, all the thing will be looking pretty white. Why is that? It scatters every single visible light that you shine on it. Very high scattering sample. In that sample, you want to do this. Your ability to say whether it is coming from here or here largely depends on the geometric path. I mean, you're talking about a geometric optics and then say that, okay, this light originating at this focus, this path, right? If it doesn't take that path, then it's not going to focus here, it's going to focus somewhere else. And your ability to discriminate and tell, hey, this light came from there is lost there. And that happens. You can actually do this for samples, at least in the brain slice, to about 20 microns or so. Any more than that, scattering overrules this part, and then your ability to detect the light, forget about this, detect any light originating from this is lost. Okay. So you don't even focus properly because it scatters, and even if you focus, somehow manage to elicit, you hardly detect anything. Because you can open it up, then you will detect everything, but then of course you lose your serious. This can be nicely overcome by, of course, the brain. You can take the small section, you can nicely see this discrimination and all that. This can nicely be overcome. This can be overcome by a method called multi-photon excitation, wherein what you are actually doing is normal one-photon excitation. You have a molecule in the ground state, shine your excitation light, you take it up to the electronically excited state, higher vibrational states, depending on the matrix element and so on. What you are actually doing, and then waiting for a few nanoseconds before it can actually relax to the ground vibrational state of the electronic excited state, and fluorescence. Right? That's what simple fluorescence is all about, right? And you detect this fluorescence. Here, in a two-photon or a multi-photon in general, what you do is that you don't come up with a light that matches the energy gap, but intentionally you go roughly about half. Why is roughly about half and all that? And not exactly half. Talk to me after the talk, I can definitely tell you. But you go roughly about half, then what you are doing is that you are hinging on the probability that the molecule will observe two photons simultaneously, two photons of roughly half the energy simultaneously. Since it's two photons of half the energy simultaneous absorption goes to the same excited state, almost the same. And comes back. If you watch carefully, think about it, until about here, where it goes to the excited state, is the only difference. After that, whether you go here through two photon or through one photon, it's exactly the same. It's exactly going to do the same fluorescence, right? And that's what your trying to orient ISP network should be fine, right? Okay, so what you're actually looking at is that the path to the excitation, that's the only difference, right? So otherwise, pretty much you get the same amount of fluorescence, same kind of fluorescence what you get with your photons. It has tremendous advantage. Number one, such a process is highly improbable. Right? We all grew up saying that, hey, if you want the molecule to get excited, match the energy. Right? We never talked about not. Okay, are we good here? Okay, we're good here. All right. So you will The molecule will absorb, but with a very, very low probability. Okay, so I just turned it to yes.
Yes, fine. Now it's fine.
All right. So, because it's a lower probability, I here talked about 2 photons, but you can actually in general go with lesser and lesser, like 3 photons, 4 photons. Basically, you are dividing the energy by multiple. And fluorescence, or the rate of emission, is some k constants times I to the power n. N here is 2, so I square. If it's 3, then 3, I cube, so on. Okay? So think of this roughly as this is a sequential event. The probability for the first event is proportional to I. So 2 have to happen simultaneously. So it's I square or n is that's your typical scattering, right? As a function of wavelength, if you think in terms of Rayleigh or a me, doesn't matter. It depends on the particle size. The brain lies somewhere in between this, the red line and the black line. For the common fluorescence, common fluorophore that we use, like a DFP or so on, you are using this, like the black arrow is. Very large amount of scattering. However, the same fluorophore, if you go by this 2 photon excitation, you are going roughly that place, half of the energy, twice the wavelength, right? The scattering is tremendously reduced, and yet you are able to elicit the same amount of excitation. So now this is super useful. Apart from this, people must, I myself have done this, and people don't know right now in my current lab, during my graduate studies, and people use multi-photon excitation for other reasons, which is if you want that absorbs in you, correct? So then shine a UV light and hope that you will be able to do a live imaging. Right? So what you do is you go with 1/3 the energy of the UV that takes you to the infrared region. You can cause the excitation that corresponds to the UV. Peacefully sit and watch the fluorescence. Again, you avoid all the damage that you could think of causing. Now this is very good. All that I explained is that you can use a different wavelength light, which is less scattering, to cause the same excitation. But how about, or how do we get the Z resolution, right? And that ability to tell things apart in depth, right? All that I have said is, okay, look, instead of using this light, you can actually go ahead and use this light, which is less compared to very, very less scattering. So you can actually reach deeper. That's good. But how about how do I get my depth resolution? That comes from the fact, right? It's my bad. That comes from the fact that I'm going to focus the light. Okay? I'm going to localize the light photons both in space and in time. Space, you guys already know. I actually have to use a lens, focus it down. So When I focus it down, what I'm actually doing is I'm taking the same number of photons, but I'm concentrating in the space, which is the area is progressively decreasing. So the number of photons per unit time, per unit area is decreasing very high number density of the photons, thereby increasing my chances of getting the good photon excitation. That itself is not sufficient. I still need to do something called as temporal focusing. I start to use pulsed lasers. Okay? The probability is so low that we need all these factors. For example, the pulse laser that we use typically in the lab is the titanium sapphire, which is about 100 femtoseconds. The light goes on and off in about 100 femtoseconds. That's the tau that we are actually talking about, the pulse width. And this happens at very high speed, 80 megahertz. So what it does is that it allows me to get this peak intensity, while on an average, I'm consuming only so much of the intensity there, the average intensity, or power there. Let me go about 10 power 5 fold more constant with all that. And that's your practical illustration of what can happen in the lab. You have the lens focusing the infrared beam in this one direction. Another lens here focusing the one photon, the blue light in this direction. That's the focus because of the one photon, and you can see the spot here, that the two photon here is pretty localized. Now that's beautiful. Now, what it tells you is that you can localize the excitation to a single spot. Then you don't have to worry about the putting in a pinhole, right? Because I know where the localization is, the fluorescent photon is coming from. I can get rid of the pinhole. I don't need it. I can completely collect all the photons that is originating here. Typically, when we talk about this, and we're going to use it for imaging, we need to be talking about resolution. Whenever we talk about resolution, first thing that comes to my mind is that what determines the resolution? The wavelength of the light. Rest of it will be constant. Here, I'm actually going for a higher weight, lower, because the spot size, the resulting spot size will be wider. The way it turns out, we can actually go ahead and calculate it's slightly wider than the one photon. Why slightly and not twice more? Because the probability of excitation goes as far. So there is a give and take. Your wavelength goes as twice, and the probability of excitation also goes as far. So if you actually do the math and look at the resolution, yes, true. The 1 over E, the distance by which the peak intensity falls down by 1 over E or E square, is slightly wider in 2 photon compared to 1 photon. That being said, what is going on here? Right? That's not what you would expect for a larger 1 over E. Right? What is going on here? What we argue is that if you actually look at the contrast function, because when you're looking at a picture like that, or even doing any imaging, what you are sensitive to is the contrast. That's what is giving you at the end, it's a ability to tell whether it is here or somewhere before. So when you look at the contrast function here, all that you have to do is to subtract 2 photons from the 1 photon. That's about your 0. So whenever you see below here, that's the extra intensities that are present here. Clearly, the 2 photon has higher. That's why you see this negative intensities that are present. However, look at this. These are the opposite sides, right? So while the 2 photon around the focus, it is exciting more of the volume. 1 photon excites a lot of fluorophores outside of the focus. This is the reason why you need this pinhole at all in the 1 photon. On the other hand, yes, it's true, it's wider, but absolutely from the outside. That's why you see this very brilliant distinction between the 2-photon and 1-photon. They're very nicely localized, but spread outward. Okay? You can actually do this. All that you are doing is you are actually trying to estimate how many number of molecules you are exciting as a function of volume. And you do this and then calculate it, and that's precisely what you do, what you get in that experiment under mathematics. That's the agreement that you see, and there is nothing more than this that's going on. So bottom line is the contrast is much better, even though the focus spot is slightly wider, giving you the ability to use this method so that we can image as a function of depth without having to use a pin. That's the key. Now go ahead and use it in your brain. It's a little bit of an involved slide, but let me walk you guys through. Because we are using a femtosecond laser and a pulse laser, what you need to do is you need to take care of a few other things than what happens in a normal microscope. Number one, you have to look at the dispersion of the pulses. Right? The fact that there are femtosecond pulses, when you actually send them through various different optical elements, they tend to disperse. So what you do is, as a result of femtosecond pulse no longer remains as a femtosecond pulse. So you can do something called as a pre-compensation. We do that using a prism pair, and then we have to be mindful about how much we have to compensate. You don't want to compensate more than what you want. So you measure it with a small interferometer here, and then let's not worry about this, but then you zoom it to make sure that you are getting focus that is not too tight and not too loose. Not to normal 1-photon full focus system, you want to put tight focus. But because of a scattering sample, to have a higher ballistic photons density, you want to optimize that tightness to meaning the amount of backfilling of that aperture. So you do that, and then you send it through a microscope objective onto the mice. That's the mice you're seeing under the microscope. And the mice by itself cannot go just directly there. It's right now anesthetized with a small anesthesia continuously flowing through. The way it needs to be done is it needs to be prepared. The way you prepare is that's a close-up shot. You can see this eyes are ointment. There's anesthesia flowing through. We shave off the skin and then make a small hole in the cranium, in the skull, and then you implant a glass window through which now you can see the beautiful blood vessels supplying to the brain. And then on top of it, we place a cover glass. And then this small contraption so that you can tighten up. You can actually hold this mice under the microscope using a simple screw system. Okay? So you can do all this. Mice survives. Not just that, it can be you have to let it recover for about a month or so. You make sure no untoward effects are there, so they happily roam around, go about doing things. Then on a fine day, we decide to make sure that we can teach them something. Okay? The idea is you want to teach them, see in the brain, in response to this teaching, what has happened. Right? So all this build-up that we have done so far is to be able to see it inside the brain. Now, we're going to go in and say, if you look inside the brain, what's going to be? Good? Everybody with me here? Yes? All right. Any questions? It's a good place. Yeah. I have a name, Balaji. Yeah.
I don't think facing a color glass.
Or cover glass, but not color glass. It's you don't want to keep it open because, as I said, I'm going to remove this cover glass. So I'm going to remove the skull, place a cover glass, let the animal roam around freely. Correct? So I can't run freely because pathogens from outside will enter, make the animal sick, and it's going to die. I have to seal it clearly. I can't seal it with anything else. I have to have the skull is impossible. I mean, I can do so many different things, but still, skull will be impossible to penetrate. Though I'm going to retract that statement towards the end of it. I'm showing how we can do that. Okay? So that's the idea. Okay? We put the cover glass so that isolate the brain. At the same time, you are able to see. Question? Okay, excellent. Now, yeah. What do we mean by that? We come down. So with this, what can we do? Number one, we cannot just image for one time, right? You use this vasculature, the structure of the vasculature. You can come back to the same place day after day. Okay? I am actually showing you this. Now this is a registration at like down to about 0.5 millimeter kind of a scale, right? But I'm talking about microns and hundreds of nanometers, right? So from here, then you just use a simple coordinate geometry to get back to the regions of interest that you have actually identified. You can go back again and again. See, day 1, day 3, day 5, and day 8. We have done that for about a month. That's the scale for about 50 microns. And this is how it looks when you look at the brain and label the neurons. Not all the neurons in the brain, but a few of them are labeled. And that's the label that you're actually looking at. You can zoom in. Okay, so that's one portion of this area you're actually zooming in. Day 1, day 3, day 5, day 8. Okay. Those are the black dots that we actually saw in the Kaha's image, right? All these times, you can actually nicely follow it through acoustics, no problem at all. With this, let's go to what we can teach to the mice and what we can see. When we talk about teaching the mice, there are many different tasks that we can actually administer to the mice to. Okay, before going there, just harping a little bit more on what we can do. You can actually do in a confocal microscope, right? That's what you can do. I mean, I don't know if you can see, down here is the cell body. That's about 600, 650 microns. The reason why you don't see much here is that all of the harbors are on top. You can actually see this long pillars. Just that our inability to express fluorescence down here limits us. Otherwise, we can easily go down to about 600, 650 microns. Just more than order of magnitude better than what you normally do with the light microscope. Okay. So now coming back to what we can teach the mice. What we can teach the mice is make the mice to understand or learn a particular context. For example, you're all here in this classroom. Right? The idea of a classroom, or idea of this particular lecture hall would be very different in each one's head. There are some common features that are embedded into the mice. Same way, if you take a mice and put it inside a chamber, it will encode that information regarding the chamber. So we can actually look at the changes that are accompanied because of this event. How do I know that it is because of that event? Because I image before exposing the mice to that event and after exposing the mice to that event. Because I can go day 1, day 3, day 5, day 8. If that is the case, I can do this before and after. Very simple. Just hold on that part to that part process. We will come back to actually give you a specific example and show you what it is. Very good. So, what will increase or what will change needs to withstand the lifetime of the plant. So it needs to make some new proteins, we have new genes. And I have to share the screen. All right. And then minimize it here. Okay, good. So we decided to ask the question of, can we first follow this at the level of the gene expression? Look at the neurons. In the neurons, can we study the gene expression? What do we mean by that? Let's say, yeah, let me share what is happening. Okay, so in response to an external signal, in response to a memory, the first set of things happen is that there are this mRNA of a gene called CFOS, immediate early gene, that gets triggered, resulting in generation of the protein, so CFOS. Being an immediate early gene, and the protein itself is coded with a degradation. So after this, which means the protein has a finite lifetime. So there is a sequential chemical reaction, and there is a limiting substrate. Substrate is E. So what is the limiting substrate? Because it cannot keep generating it all the time. Then the memory is the response to the memory event is not there. So that's possibly maybe kind of said, okay, this could represent simple gene expression kinetics following memory. So that's our hypothesis. So if that is the case, then you can actually go ahead and solve the simple chemical reactions, chemical equations, the rate equations for representing the chemical reactions. Then when you do that, what you are actually looking at is that if, for example, I could generate this protein together with a fluorescent tag, okay, then I should be able to visualize this protein and then say when a memory event is. That's our idea. Okay, so the kinetics of the B should be given by this, right? It's just simple rate equations, and you can solve the couple rate equation, you get that. Very, very simple rate equation. And translate into fluorescence, very simple. What you do, that's the number of molecules, you just multiply efficiency. Fluorescence of time. You plot it. Okay, so of the exponentials. As a result, you see one of them going up and then the other guy, right? It's a reverse exponential 1 minus e power k. This guy is e power kd. So you add them both. So what you get is this peak. Now, the peak where it happens is tied to when the event has happened. This red If there is an event 2 that comes a little later, we are going to see this. We need to make sure our event that happens in mice's life is very salient to just that. Yes, the way you do this is you shut down everything that the mice can kind of think of that as a normal. So the 1 month period that I was talking to you guys about, part of it is this, where nothing new happens in this mice's life for that period of time. For about a week, if you continue to do this, whatever you're doing routinely, you don't miss anything, you don't add anything, you just keep doing this. We call that as habituation. It happens to all of us. We are all here in Bangalore, beautiful, nice summer, but it's not too hot. And being in Delhi or in Rajasthan, temperatures are so high, you can't probably think of wearing a cloth that we are wearing here. Right? You'll be wearing something else. Same is the case with the winter. When you change, right, the weight of the cloth changes dramatically. Your winter clothes are not as light as your summer clothes. Your body doesn't feel it. Very first time, if you notice the end, when you try to sweater them up, big body, right? They don't like it. So out of many different reasons, one of them is they're not used to. This not used to is what this habituation is. Used to the fact that they will be inside the proteins, nothing happens to them. Repeat, no genes gets expressed, so that no new event per se has happened. After doing that, we take them and put them inside we put them inside the chamber and then provide them a okay. Share. And the next time, when you actually bring them into the same chamber, they express this uneasiness by seizing every single moment. They freeze. That's a good indicator of, okay, they do remember this particular context. Okay. So in response to this memory event, you are actually looking at the expression of the genes. These dots that are the neuronal nucleus, right? Here you don't see this beautiful process. The gene expression happens in the nucleus over L. So that's why you are actually looking at these spots, which are basically the gene products of and then select this. The mice's brain, right, as a function of time. The red, the fit to the expression that I have derived, okay, and you can see many different cells. These are numbers. Okay, the examples of few examples of the cells that we have given, and you can beautifully follow these events, clearly telling roughly about this is the peak, right? So that from the peak, you can know when the 0 time is. You can actually extrapolate to know when the event has happened. You can do that for every single cell, get a beautiful histogram of the K, thereby identify where exactly the event has happened. But the beauty is not just identifying where the event has happened, but you can also tell which of the neurons are actually encoding which information. With this, what I'm going to do is, I was talking to you guys about this chamber A, with one chamber and giving a mild foot shock. You can start to ask multiple events. Right? We are talking about comparing multiple memories, right? So if you have multiple memories, can I identify multiple populations that are encoded? Okay, the way we do that is chamber A, foot shock, 24 hours later, chamber B. The symbols are different because the chambers are different. The features are different. Then you put them back. This corresponds to A, where they got the shock. B, they don't have a shock. So that's how you differentiate. Mice also differentiates. Then you can go ahead and image them following this and some little bit of a time differences. The whole idea is that you get a series of these cells. From there, some of them represent the first cell, one event, some of them, and these guys correspond to the second event cells. And I don't have multiple event cells examples here, but you can do this, and then the portion of the brain you reconstruct and then show wherever there is overlap. They responded to both these.
What is there? Yes, always there, but sometimes.
Okay, so roughly about 60% of the cells that we actually looked at in the area were getting activated following the first memory, and a little bit more than the 60% for the second. But I don't think we can differentiate them. But the key point here that I would like you to note is that in a given region, which of the neuron is actually encoding memory is completely random. And if it is random, and we are talking about two such memories, then the ensemble that represents both of them should be a few chunks. This is 60 of 60, right? 60 times 70. Right? That's roughly what we see. The overlap fraction is just about few chunks. Okay? So there are two different ensembles. There are two different memories. You can actually trace them, and I am a little bit over in time, but I also started late. Maybe about 5 minutes. All right. So now, what I told you, I talked to you about this, right? Ensemble, these neuronal ensembles being able to see this. I'm going to show you a beautiful conceptual different idea that came because of the multi-photon microscope, right? Until now, when you talk about plasticity, all that you talked about is, all that we know until now is that, okay, you can change the threshold, you can change the throughput, you can change some molecules or the network connectivity, right? The increasing more number of spines. What I'm going to talk to you about is that there is a new way of thinking about plasticity. For this, what we did was, again, the same thing. You can actually see this beautiful 3D rendering of neuron. Just look at this. You can actually nicely see this synaptic connections, putative synaptic connections, the spines. Here, the labeling is different. We are actually labeling the entire neuron, unlike the previous one. So what you're going to do is we're going to talk about these spines as a function of memory A and memory B. These are the two same memories where we saw the populations are only random chance overlap, right? But could there be a relationship that is established between them? That's the question. So in order to do that, we looked at the spine density. Okay? And when you look at the spine density, whether the first memory or a second memory, following a new memory, right? The spine density marginally increased, no doubt, but the second memory did not increase any further. Okay? If you were to keep adding more memories, spine density were to increase, you should see this. There is a problem there, right? How much can you keep on increasing? At some point, it will be a cube, smooth cube, right? So that cannot happen, right? So what is going on here? How do we actually store multiple memories that are related? So in order to do that, we wanted to test this hypothesis. Okay, look, you can do the information in a way. When you talk about spine, increase in density, you don't have to increase the density throughout the dendrite, but you can actually reorganize the spine, right? If you look at the entire dendrite, the number of spines are exactly the same, but what has happened is that this spine, instead of being here, is moved here. That by itself can actually encode information, and if it was this, the metric was very simple. We said, okay, look, this is like a long knots here. So zero from the sun autopolar ground. You are actually asking how similar is the local structure across the dendrite. The idea is if they have come together, you see the similarities staying for a longer period of time. Okay, so there will be an average similarity that's about decaying in a little around this point, and this is a short scale similarity. You think of this as short scale correlation versus a long scale. Long scale talks about the density, while the short scale talks about the clustering. Kind of giving you as an information, not providing you an evidence, but that's okay. You can talk to me later. Okay. So when you do that, the long story short, the bottom line is we saw. Sorry, my bad. What we are actually able to see is that the clustering of the new spines are different, right? When you form a second related memory, what is happening is that the arrangement of the spines rather than the spine density is changing. That's the bottom line here. We can actually go ahead and test it. How we test it is you do some transgenic mice go and activate those neurons, right? Now we observe. Until now, what we have done is observe, passive. Now what I'm going to do is there are optically activatable channels, ion channels that are present. I'm going to express them in the mice, make the mice undergo these tasks, right? That's the training context here. And that's how the brain looks like. And then through optical fiber, actually shine the light, make the animal evoke that memory in a completely different context. Okay? The answer is that, yeah, you can actually make use of this and say that, okay, that this is sufficient to actually cause the memory. Okay. I will just stop there. We also make some devices to measure it in humans, but that's besides. Thank you all.
So I would say, very absorbing talk, great analysis, depth analysis I could see, and the arrangement of spines is something that I have heard for the first time. Yeah. So that's, you have some questions. Okay. Fine. I do have some questions that I thought I will, do we have a way to manipulate memory by external means? Like to, to delete a kind of, memory or to delete.
So you can do that. What you can do, you can subtract. Right here, I gave an example. I showed an example of how to make the memory express, right? By shining the light in a completely different context. The mice are trying to remember A. I'm putting them in a context C and making them feel that it is actually A. You can also do this, which is by expressing a different kind where it inhibits the neuronal activity. You can make the animal put back in. You can put back the animal in A and make the animal think it is not A by shining the light. So that's one way of manipulating. Behaviorally in humans, I mean, this is happening in mice. Behaviorally in humans, that's not something nobody would like to have the 5% but you can do some few other tricks where you find them in relation to the other. Those are typical psychiatric gimmicks to manipulate.
One of the things, whatever we do in mice, does it directly translate to humans?
Yes and no. Many of them do, and some of them don't. Overall, the basic understanding you can draw, definitely. But then if you actually want to completely translate by dot, there are basic problems. One is that you don't have a parallel task running between the mice and the humans. Mice are white, humans are humans. It's not and all that. Towards that direction, taken a few steps where we are trying to see if we can construct a task that is across the humans and across the studies. That's one limitation. The second limitation is they are different organisms. They're bound to have differences, and we have to be cognizant about that.
Finally, I have one more thing. Like we talked about sacrificing modes.
Yeah.
So you do a certain kind of experiment and sacrifice and try to analyze. Does this sacrifice not affect the state?
So these are not the sacrifice. These are all irreversible. What I have told you. What I said was before the advent of this, the only way you can do, even now, some of many other things, is to be able to sacrifice the mouse and then see that. But to address your question, does it probably no, because these things take time. Any effect in the brain takes 40 minutes before something can trigger in. So if you are quick enough to extract the brain and do your analysis or fix the brain, then you'll be fine.
So there are no questions in this. Solar is fine. The brain stores some function. It is rearranged. Right. It's not
Making new. Yeah. So great question. So what happens is that this is only for A and B. They are related. So you have a prior memory. On top of it, you are trying to store the other memory. So it's so pruning of this, what we talking about the pruning, refinement of the previous memory to encode both of them. Is what we are showing. If you completely bring out with a different memory, like a C or D or E, correct? Further away from the initial memory, this won't happen. That's why we test them, whether it happens behaviorally or what you are overwriting or not. I haven't shown here, but in our lab, what we could show is that you have the first memory and you are storing the second memory, right? So what we call it as now, the second memory is not necessarily a new memory all by itself. It's a memory that is integrated to the E. So if I can actually try to extinguish this memory, it should have hit up. While I extinguish this memory, it should have hit up. You see the beautiful this dissociation between extinguish this state. There is a change. Correct. But the events are happening constantly. I'm not sure about the feedback to tell whether the events is changing or not, but events are happening. Whether you store it or not, events are happening. Okay, so there are mechanisms to that release a kind of modulators or neurotransmitters that enhances that enforces that this information should be stored, while the others can be kind of overlooked. I mean, others overlooking is a different issue, but for sure, emphasizing the fact that needs to be stored is driven by some modulators. Neuromodulators can actually facilitate that. Event happening, we don't have control, correct? And that is continuously happening. Your brain is not. Your brain is keeping track of as much as what it can, but that it doesn't have a control over happening. As a result, it doesn't tell it has stopped.
When you're saying that memory is like our brain is not so it means that I was saying about that synaptic nerves are not forming, but that connections are not.
Yeah, that's one way of doing it. Right? Just the way plasticity was talking about in multiple scales, right? From a network to the synapse. This can also happen at the multiple levels. But more interestingly, one of the very prominent one is forming a new connection to inhibit the old one. So inhibitory connection. I was completely paying attention to the XLFU. Good part of the lab also studies an inhibitory process where our brain develops the habit, develops a way to inhibit certain existing things. That way, you can actually focus.
Okay. Last question.
The subconscious level, as so peak. We would like to classify it as general. We call it as declarative. Okay? Declarative here is that me being consciously able to recollect something. I ask a question, what is your name? You recollect something and tell me that, right? So that we call it as a declarative or consciously able to retrieve that name. There is also a non-declarative memory. These are not necessarily requiring a conscious. That's what you mean by subconscious. Like, for example, you get onto your bike, right? You don't really think about my right foot is high, right? That comes, I mean, in fact, if you start thinking about it, you will fall down. You don't want to do that. These are procedural memory. Procedural memory is one kind of is non-declarative memory. There are other memories too. Reflexive. You can develop reflexive. Those are also non-declarative memories. They are occurring in different regions of the brain. The regions of the brain that is declarative memory technique is of interest to me. That's what I'm studying. All the events that are happening in our lifetime, they take the representation in this way. Yeah.
So maybe you can talk about it. So, yeah, you know, we don't know if you wondering. It looks like there is a lot of interest. So, you know, I did not ask him, but maybe you can visit that.
Yeah, please.
And, yeah, I mean, so much of interest I see. So, you know, the visit is there. But anyway, thanks, Balaji. Thanks a lot for it.
Thank you. Yeah, please stop by. We are just stone throw away, and just email us, and we'll be more than happy to chat anytime about memory or about microscopy.
You know, thanks.